Okay. So next we have Christine Rushlow. She's going to tell us about Zelda.
Oops. Okay. Today I'm going to tell you about the zinc finger transcription factor Zelda and how it pioneers early enhancers during zygotic genome activation. The work that I'm going to be telling you about was done in collaboration with Julia's lab at the Stowers, and I'll just begin with a very quick introduction to early development. Okay, so Zelda plays a key role during the first 3 hours of development when these important processes are taking place. This is the time when the maternal to zygotic transition takes place, where we have the degradation of thousands of maternal RNAs and the activation of zygotic gene expression, which starts in the second hour with about 150 genes being activated, and this is followed by a major burst of transcription in the third hour when the process of cellularization takes place during that long interphase of nuclear cycle 14. So the transcriptome is changing very rapidly and very dramatically during this time, so it's a major reprogramming event. So from the work of several labs, including mine and the labs of Tom Klein, Eric Wieschaus, Melissa Harrison, and Mike Eisen, we know that Zelda binds specifically to CAG-TAG and related sites. We also know that Zelda is uniformly distributed in nuclei very early, and in fact, these embryos up here, showing embryos in different nuclear cycles, are stained with anti-Zelda antibodies. Okay. We know that there are 2 types of Zelda targets. The first type of target are genes that are expressed very early. They're expressed genes, for example, that are involved in processes such as sex determination and cellularization, and also other early blastoderm genes, such as the gene HALO, which is expressed ubiquitously like Zelda, and is mostly absent in Zelda minus. The second type of target gene are the patterning genes. And these genes, as you know, are not expressed ubiquitously, but expressed in discrete spatial domains because they're regulated by the patterning morphogens, such as bicoid, the anterior morphogen, and dorsal, the dorsal ventral morphogen. For example, high levels of dorsal in the ventral region activate genes like twist and snail, which go on to form mesoderm. Lower levels of dorsal, here in the lateral region, activate genes like, for example, rhomboid, which we heard about earlier, SOG, and Brinker. And we know that Zelda plays a role in regulating these genes, because in the absence of Zelda, we see effects, for example, of Brinker. This is showing that lateral stripe. And in Zelda minus, that's, first of all, transcription activation of Brinker is delayed by 1 or 2 nuclear cycles, and you can see that when it is turned on, the pattern is reduced to a very narrow and sporadic stripe. And so we think that dorsal, which has not changed in the absence of Zelda, where it is at still relatively high levels or intermediate levels, it's still able to activate target genes like Brinker, but when that in the lower end of the gradient, now dorsal is no longer able to activate target genes like Brinker, so we think that Zelda somehow potentiates dorsal activity in regions of low-level dorsal. I'll come back to this in a minute. Okay, so there's many genes that fall into these 2 types of Zelda target genes, and so we refer to these as class 1 and class 2 genes. And one question that's been bugging me for quite a while is, why can't Zelda actually activate class 2 genes ubiquitously, like it can activate class 1 genes? And so, for example, it could be all about repressors. We know that many of the GAP genes, for example, are repressors. We know for the DV genes, the neuroectoderm genes, snail in the ventral region acts as a repressor. But I think there's a little bit more to this, and that comes from observations made by Kai Chen and Julia Zeitlinger, and also some experiments we did together. of certain features of these classes of genes. For example, we know that Pol2, when we do Pol2 chip at these genes, that Pol2 is absent in Zelda minus. And we also know that class 1 genes have promoter motifs, they're enriched in TATA and Zelda binding sites. And so we think that Zelda basically binds just upstream of the transcription start site at these genes, recruits polymerase, and has a direct activating role. Now, for class 2 genes, we know that Pol2 is reduced but not absent in Zelda minus. And for example, for the dorsal targets, these genes, like Brinker, are actually Pol2 is absent in dorsal minus along the gene body, and that these genes, their promoters are enriched for motifs that are different from the class 1 gene motifs, promoter motifs, and they include, for example, initiator, MT, GAGA, DP, and pause button. So we think what's happening here is that it's possible that Zelda interacts with these promoters that have these motifs, including TATA, and of course the Zelda binding sites, but it can't interact for some reason with class 2 gene motifs. And so we don't know the answer to this yet, but we're doing promoter swap kinds of experiments to try to get at, possibly change a class 2 gene into a class 1 gene. So what basically is Zelda doing then at class 2 genes? And so we think that it's playing more of an indirect role, and that, for example, for dorsal targets, dorsals interacting with the promoter, recruiting polymerase, and that Zelda plays more of a potentiating role of dorsal or other patterning factor activity. And we think that, or we have some evidence that Zelda basically is involved in helping these other transcription factors bind. For example, if we look at dorsal and Zelda chip by ChIP-seq, we can see at the Brinker enhancer, for example, that Zelda and dorsal bind to the motifs in wild-type embryos, but in the absence of Zelda, we see greatly reduced dorsal chip binding at these regions, and we also see this at other dorsal target genes. Now, so we think again that dorsal is helping Zelda is helping dorsal bind. Since it's also been shown that Zelda can help bicoid bind by Steve Small's lab, and Alex Stark's lab has shown that Zelda can help twist bind, we think that Zelda's playing a more general role in helping transcription factors bind. And so we can ask the question, does Zelda play a pioneering role? In other words, as Justin mentioned, getting into chromatin, binding chromatin, opening up chromatin for other factors to bind. And so we can ask the question, does Zelda open chromatin? And we can also then ask, is this a mechanism to prime enhancers globally during zygotic genome activation? So I'll address the second question first, and I'll turn that question around to something we can actually look at and ask, are Zelda-bound regions enriched for early enhancers? To answer this, we took collections of enhancers that have been experimentally verified, and we looked through those for early enhancers. And we came up with a list of about 1,000 enhancers. So again, regions of the genome that have been shown to drive reporter expression in early embryos. And then we asked, are Zelda-bound regions are enriched for these enhancers? So now I'm going to show you Zelda-bound regions. So this is just Zelda chip data. This is just a heat map showing Zelda-bound regions from strong to weak, all aligned at the peak center. And you can see that there's a nice correlation with the presence of Zelda binding sites, motifs. And this column here just shows every red dash is one of those enhancers, 1,000 enhancers, and about 70% of those enhancers actually overlap with the Zelda-bound region. And you can see that the stronger the binding, you have an enrichment for those enhancers. Now to get at the other question about can Zelda open chromatin, so we've started to analyze that, and so we took the approach of actually looking at nucleosome occupancy, and so we used the technique MNA-seq, And microcalconuclease at the right concentration will digest DNA in between nucleosomes, leaving then the DNA wrapped in nucleosomes. And that's what we actually sequence. And so what we do is MNase digest chromatin from wild type and Zelda minus embryos. We gel isolate mononucleosomes, sequence the DNA. And then we assess, what I'm going to show you is assessing nucleosome occupancy at Zelda-bound regions. Okay, and I'm going to show you that data in the form of a metaprofile. And so what we're looking at here now is the top 1,000 Zelda-bound regions which are enriched for those early enhancers. So basically, we're looking at enhancer regions. We're aligning them at the Zelda summit. And we're looking now at MNase reads. So when you have high MNase reads, that means that you have high nucleosome occupancy. So from wild-type embryos, that's shown in blue, and you can see that around the peak summit, we have lower nucleosome occupancy than outside regions. And notice that in Zelda minus, we now have higher nucleosome occupancy. So that means now you have more nucleosomes at these enhancers in the absence of Zelda, whereas in the presence of Zelda, you have less nucleosomes. Now, let me just point out this gray line here. This gray line is a prediction of whether the underlying DNA sequences at these regions favor nucleosome occupancy. So again, this is just a prediction algorithm from Shi et al., and we're just asking whether these DNA sequences actually favor nucleosome occupancy, and so you can see that they do. And so we're making then the extension that at these regions, which are enriched in enhancers, they prefer to be occupied by nucleosomes. In other words, nucleosomes will form around these sequences. So then this tells us two things. This tells us that there's an intrinsically high nucleosome barrier at enhancers, and that Zelda lowers this nucleosome barrier. Okay, so our current model then, which looks very similar to what Justin showed, is that before zygotic genome activation, when we think that Zelda levels are actually at pretty low, we have a nucleosome barrier. Zelda comes in, and it will actually displace or eject, we don't know this yet, nucleosomes, and then therefore imparting enhancer competence. Then when you have other transcription factors, they can come in, and together now with Zelda, they can, via nucleosome-mediated cooperativity, kick out and eject nucleosomes and then enhancer activity will follow. Okay, so although this is our model, I just want to point out that we don't know the status of chromatin in early embryos. It is possible that chromatin can be relatively open during these rapid nuclear cycles. Also, as Shelby showed this morning, certain open regions are maintained even through mitosis, and so Zelda could just be opportunistic and get in there before anybody else and then keep things open. And so either way, we still think that it's actually helping other factors bind by doing this. Okay, so I just want to end by thanking the people in my lab who did this work. So Lydia, Joey, Mavis, and G did the work that I showed you today. Sandra and Nikolai are actually working on how Zelda zinc fingers bind DNA and what sequences they bind, and Nikolai has a poster that I've listed there. And I also want to thank Kai Chen, who was a graduate student in Julia's lab, but now is a postdoc with Mike Levine at Princeton, and Jeff Johnston and Julia for this really wonderful collaboration. Thank you. Shelby.
Yes. So for the nucleosome positioning with Zelda and Zelda minus and wild-type embryos. So if you took a random selection of Zelda binding sites in the genome and plotted the nucleosome occupancy, particularly the ones that are not remodeled by Zelda, do you see a similar distribution or prediction? Or is there something about the way that Zelda motifs are arranged around nucleosomes specifically that makes them sensitive to disruption by Zelda?
Yeah, so we basically looked at Zelda-bound regions. So if you just go for, you mean Zelda motifs and just align regions by Zelda motifs, we haven't done that, but I don't think that's the case, because even if you go to weaker Zelda-bound regions, you know, basically the difference in the nucleosome occupancy becomes less and less. That's why we focus on the top, the most strongly bound Zelda-bound regions. So, yes?
I think I have a very basic question. At the beginning, it seemed like you were talking about Zelda binding or having a relationship with these other transcription factors, like dorsal. But then at the end, it was more like Zelda opens up and then transcription factors come in. So is there an egg and chicken kind of thing where it has to be an interaction between the transcription factor and Zelda before Zelda can actually open up the chromatin, or Zelda opening the chromatin has to come first?
Okay, so we think Zelda plays this pioneering role, which by definition, it gets in there first. I mean, for dorsal, you know, dorsal has to go through this regulated nuclear transport, and even by antibody staining, you can see Zelda is in there, you know, after telophase before dorsal. But I don't know about Vicoid, and I don't know about the other factors. And we also don't know if Zelda can actually stay bound. We don't think so. We don't see it by antibody staining, but that's the only thing we've actually done. So yes, we do think that it has to get in there first. And we don't think there's like protein-protein interactions specifically between dorsal and Zelda and Bicoid and Zelda, but that hasn't been ruled out. We haven't actually tested that.
Thank you.
So I hope that answers your
One more question.
Yes, so
Very
I guess
quickly.
relative to your alternative hypothesis, and that is anything known about how quickly Zelda binds after replication in the process of chromatin reassembly, right? I mean, and then the other thing is it bound during mitosis, because, you know, it could be doing this process known as bookmarking, that is keeping the promoter open rather than generating it, right? Which is also interesting.
Okay, so very quickly, so again, we've only looked by antibody staining, and even during telophase you can see it, but then before that we don't see it in there by antibody staining. Melissa Harrison has actually looked with a live GFP Zelda, and she does not see it bookmarking either. And the other question...
Replication.
Oh, replication.
Yeah.
And so, yeah, that was the same thing. And also, I'm going to walk off now, but Jerry reminds me that nucleosome-mediated cooperativity was actually first hypothesized by Jerry.